Hi, good morning, good afternoon, everyone, depending on your time zone. So today we're going to have the next round of the Mnemonic seminar series, and Dr. Leticia Festito from my team in Queen Mary University of London is going to give a presentation on, well, basically an introduction to variant prioritization. So with that, I think I'll just hand over to Leticia. Yeah, and please go ahead.
Hi everyone, thank you for having me here today. In this session of the Monarch Seminar Series, we'll be focusing on variant prioritization, which is a key component in the interpretation of genomic data. So this is a quick overview of what we'll cover in today's seminar. We'll start familiarizing ourselves with the concept of variant prioritization, what it means and why it matters. Next, we'll walk through a standard whole genome sequencing filtering pipeline and discuss some of the key changes associated with it. Then I'll guide you through a comparative overview of several phenotype-driven variant prioritization tools, highlighting how they differ between each other. And from there, I will talk a little more in details about Exomizer, and we'll move into a real-world application by showing our recent reinterpretation project within the 100,000 Genomes project, where Exomizer played a key role in solving previously undiagnosed rare disease cases. So let's begin with what variant prioritization actually means. At its core, variant prioritization is the process of identifying and ranking one or two disease-causative or clinical relevant variants from among millions of variants in a person's genome. This task is often compared to finding a needle in a haystack, where the needle is a rare segregating pathogenic variant responsible for a particular trait or disorder, and the haystack is the vast background of a benign genetic variation present in every individual. And as you will see through the whole talk, this is absolutely crucial to enabling an accurate diagnosis. So here I would like to show how a standard whole genome sequencing analysis pipeline for rare disease usually looks like, and discuss some of the key challenges associated with it. These figures represent the initial step, where we begin with over 6 million genetic variants per genome, which are here depicted as stars along these chromosomes. The first step in a standard whole genome sequencing analysis pipeline for rare disease usually involves excluding variants that are commonly observed in the population using frequency filtering. We begin with over 6 million variants per genome and initially prioritize those that are rare based on allele frequency thresholds derived from population sequencing databases, such as NOMAD, 1,000 genomes, and local frequencies. So while allele frequency filtering is a crucial first step in narrowing down rare variant, it actually comes with a major challenge, which is that the frequency databases that we often use, such as NOMAD, are often heavily skewed towards individuals of European ancestry. Despite efforts to aggregate and integrate data from sequencing projects with more diverse ancestral backgrounds, many of them still remain underrepresented, therefore limiting our ability to accurately interpret variants in these individuals. The second step of a standard analysis pipeline typically focuses on filtering for coding variants that are predicted to directly disrupt the protein sequence and prioritizing those predicted to be pathogenic using in silico pathogenicity predictors such as CAD, REVEL, alpha-missense, Splice AI, or RAM, which helps distinguishing potentially deleterious variants from those likely to be benign. But what are we missing by applying this kind of approach? One of the answer is a lot. Of course, one advantage of this strategy is that by focusing on a small proportion of the genome, let's remember that the exome basically represents only 1 or 2% of the whole genome, we can actually achieve a genetic diagnosis in a substantial number of cases while keeping the number of variants to review at a manageable level. However, despite the power of this approach, a significant proportion of patients remain undiagnosed after whole genome or whole exome sequencing. In practice, most analysis pipelines, particularly in clinical settings, are not routinely designed to pursue diagnosis involving non-coding regions. This is largely due to the problem of having overwhelming numbers of variants to interpret and validate. Yet there are growing evidence suggesting that a substantial proportion of the unresolved rare disease cases might be caused by variants in non-coding and regulatory regions, including enhancer, promoters, untranslated regions, or non-coding RNAs. Similarly, many unsolved rare disease cases are thought to involve structural variants, either alone or in combination with single nucleotide variants or indels. However, detecting structural variants using short-read whole genome sequencing is technically challenging, and these methods often struggle with limited resolution and imprecise breakpoints, making it difficult to confidentially identify and interpret the functional impact of a structural change. However, long-read sequencing offers a promising solution, showing an improved ability to better resolve structural variants. So as we will see later on in the presentation, many variant prioritization tools do not yet support the analysis of non-coding variants or structural variants, leaving therefore a significant proportion of the genome unexplored. Next, we filter for variants that segregate consistently with the disease in the family, following the expected mode of inheritance. This includes evaluating linkage data, retaining variants that co-segregate with disease across extended pedigrees in multiple affected individuals, or identifying de novo variants in trios. Variants that do not segregate with the disease within the family are excluded, therefore helping narrowing down likely candidates for diagnosis. However, this approach introduced its own challenges. For example, the misclassification of affected status, incomplete penetrance, polygenic or digenic inheritance, or mosaicism can all complicate the interpretation and lead to missed or incorrect diagnosis. In particular, not all phenotype-based variant prioritization tools currently supports the option to account for incomplete penetrance, which inevitably increases the number of candidate variant per case. The next filtering step is likely of particular interest to this audience, and it involves the deep phenotyping of patients' clinical features into standardized formats like the Monarch's developed human phenotype ontology or the phenopackets, and comparing these to phenotypic profiles of non-human diseases and model organisms. This includes using resources such as human disease, such as OMIM, Orphanet, ClinVar, or ClinGen, as well as functional evidence from model organism databases such as the MPC, zebrafish model, or protein-protein interaction networks from strings. So a number of phenotype-driven variant prioritization tools are actually now available and freely accessible. And later on in the presentation, we'll take a closer look at some of these tools. These filtering steps are often highly effective in ranking the causal variant among the top candidates and enabling a genetic diagnosis in a good proportion of cases. While phenotype-driven variant prioritization is, of course, a powerful strategy, it also presents important challenges. For example, is effectiveness can depend on the quality and completeness of phenotypic data, which might be inconsistently recorded in some clinical settings. In addition, a majority of the variant prioritization tool rely on the curation of the latest disease gene association by OMIM and Orphanet and the associated phenotypes by the HPO team before this knowledge is even available in the software. So this might be causing some delays between the publication of new findings and their integration into the databases. And additionally, many prioritized variants might still fall into the category of variant of unknown significance, where the clinical impact remains unclear despite the gene disease association or phenotypic similarity relevance. This step shows the importance of interoperability across data, knowledge, and systems, an area in which the Monarch Initiative has played a leading role by integrating information from numerous diverse data sources across diseases, genes, phenotypes, and species into a single knowledge base, with ontologies being used to unify across these sources. These are some of the tools that have been developed, and we'll encounter some of them again later in the presentation. So as the field of variant interpretation continues to advance, one of the greatest challenges we face is keeping pace with a rapidly evolving and dynamic environment. One important but often overlooked challenge in variant interpretation is the dynamic nature of patients' phenotype. While clinical features can evolve significantly over time, phenotype descriptions are typically captured as a static snapshot at the time of recruitment. As disease progresses, new symptoms might emerge that are more diagnostically informative. Therefore, relying only on the phenotype annotation at the time of recruitment can limit the effectiveness of phenotype-driven analysis and might contribute to delays in achieving a diagnosis. A key area of progress in the variant interpretation field is the effort to better integrate and synchronize structured phenotype data with electronic health records. One example is the adoption of PhenoPackets, which provides a standardized structured format for describing patients' level phenotypic features and that are specifically designed to interact with the electronic health records system, enabling more dynamic, longitudinal capture of phenotypic data as it evolves over the course of the clinical care. Two other of the most significant challenges facing the variant interpretation field today are the continuous influx of novel scientific publication and the rapid pace of gene disease discovery. High throughput and lower cost sequencing technologies have enabled the widespread integration of whole genome and whole exome sequencing into clinical practice and driven the rise of large-scale sequencing initiatives. These advantages have led to a discovery of hundreds of novel gene disease associations per year published in peer-reviewed journals. Therefore, staying current with this rapidly expanding body of knowledge is a major bottleneck, and new finding might take months, even years, to be curated into the reference databases. Therefore, bridging this gap remains a key challenge for improving diagnostic yield and patient care. Finally, the rapid proliferation of novel tools and databases presents both a big opportunity and a big challenge. These vast amounts of generated genomic and phenotypic data have created both the demand and the opportunity for a computational solution. And progress in machine learning and AI have also enabled the development of increasingly sophisticated algorithms, ranging from improved pathogenicity predictors to new phenotype-driven prioritization softwares. However, the performances across tools can be challenging to compare and often not reproducible, largely due to the absence of standardized empirical benchmarking frameworks. And that's why tools like FEVOL, which is a benchmarking framework for phenotype-driven variant prioritization, introduced by Jasmine Bridges in the previous seminar, represents an important progress in the field. So, as previously discussed, numerous phenotype-driven variant prioritization tools have emerged over the past decade, using different methods like natural language processing, machine learning, and artificial intelligence, including the deep neural networks, semantic similarities, and statistical probability approaches such as likelihood ratios. This table summarizes a selection of peer-reviewed, freely available tools. These tools differ significantly in their features and capability. While most tools support single nucleotide variant analysis, only a few of them are actually capable of handling both single nucleotide variant and structure variant, and even fewer of them address non-coding variants as well as coding ones. They also differ in terms of licensing, support to the Build 38 human genome assembly, family-based analysis, and programmatic access, which are not consistently implemented across all of these platforms. A further point of differentiation lies in the sources of biological evidence used. Only a subset actually incorporates functional data from model organism, protein-protein interaction, or pathway analysis. As mentioned before, the majority of the tools rely on the HPO curation of homing and orphaned diseases, and these can create delays between when the variant was described in the literature and when it actually gets added into the databases. And to overcome this, for example, Emily software offers the advantage of natural language processing of the latest literature to identify reference genotype to phenotype knowledge. Given the ongoing challenge we have previously highlighted, which is keeping pace with a rapidly evolving and dynamic environment, the update frequency of this software and the long-term maintenance of them become critically important. However, in this table, you can see that only a handful of tools, including Exomizer, AI Marvel, and Lyrical, show evidence of ongoing maintenance and databases updates, whereas many freely available tools are not maintained beyond publication. So from this list, Exomizer stands out as one of the most comprehensive and well-supported phenotype-driven variant prioritization tools. It supports both low and high throughput access, is compatible with the Build 38 genome assembly, and accommodates family-based analysis. It also enables the joint reprioritization of both single nucleotide variant and structural variants, and through its genomizer component can also evaluate non-coding variants, including enhancer, promoters, untranslated region, and deep intronic variants. Importantly, Exomizer also integrates diverse biological evidence such as model organism data and protein-protein interaction networks to support gene prioritization. This figure below actually shows the timeline of Exomizer software and database updates since 2019. The upper portion illustrates the growing number of curated human diseases, mouse and zebrafish annotated genes association in the underlying databases. And the lower portion highlights key features enhancement across software versions. So here you can see that important development include the integration of CleanVar, the ability to support structure variant analysis, the possibility to use phenopackets, and last but not least, the implementation of automated CMG classification, which is a classification system that provides a standardized framework for assessing variant pathogenicity based on multiple evidence type, similar to the ones that we mentioned before, like population frequency, functional evidence, pathogenicity predictors, and segregation. So what is Exomizer? Exomizer and Genomizer are two phenotype-driven tools for variant prioritization developed by Professor Damian Smedley and Dr. Julius Jacobsen with colleagues from the Monarch Initiative. The difference between the two is that Exomizer usually, as a default, focused on coding regions, while Genomizer extends this analysis to the entire genome, including non-coding regions. Starting from a set of clinical phenotypes encoded as human phenotype ontology terms or phenopackets, and a patient's VCF file from whole genome or whole exome sequencing, Exomizer is able to annotate, filter, and prioritize likely causative variants. So it is mainly constituted by two scores. One is the variance score that utilize minor allele frequencies from local and population sequencing sources, in silico predicted pathogenicity, variant molecular consequences, and segregation across affected and unaffected members. And the second score is a phenotype score that is obtained from the semantic phenotypic similarity between HPO of input describing proband's phenotype and the phenotypic profiles of known HPO annotated human diseases, for example, from OMIM or Orphanet, and other phenotype ontologies like mouse or zebrafish associated with the gene. So from, for example, from IMPC or ZFIN or nearby neighbors in a protein-protein interaction next from string. These two scores are then combined using a logistic regression model to generate a final combined score that is used for the actual ranking of the candidate variants. So now that we know what Exomizer is, I would actually like to move on to an example of a real data application by showing you our recent reinterpretation project within the 100,000 Genomes project. So what is the 100,000 Genomes project? Is basically a UK government project launched in late 2013 and coordinated by Genomics England. The project aims to apply whole genome sequencing to the study of rare diseases, cancer, and infectious diseases in a national healthcare setting. So after ethical approval, National Health Service patients were recruited to the 100,000 Genomes project through one of the 13 genomic medicine centers all around UK. And this dataset includes around 35,000 rare disease families. So to understand what we did in our analysis, it's important to understand what is the standard or original Genomics England diagnostic pipeline. Basically, they developed a tool called the Panel App, which is a publicly available platform to create, review, and share diagnostic grade gene panels for rare disease analysis. It uses a crowdsourced model where clinicians, researchers, and geneticists evaluate gene disease association using a traffic-like system, and they classify the gene based on the strength of the evidence linking them to the disease. This could be, of course, red if there is a limited or no evidence of the association, amber if moderate, or green if there is strong evidence of the gene disease association, and therefore the gene is suitable for clinical diagnosis. So the original 100,000 genomes project pipeline is therefore panel-based and driven by clinical indication of which panels to apply to each one of the patients. This pipeline flags for diagnostic purposes only rare segregating protein truncating, protein altering, and the novel variants affecting green genes in the applied panels. And therefore, only these variants get assessed by the clinical scientists according to the ACMG classification. And the variants that are considered pathogenic or likely pathogenic then get reported and fed back to families. Subsequently, clinical scientists also began reviewing the top 5 exomiser candidate variants per each one of the case, and this combined approach has led to an overall diagnostic yield of approximately 25-30%, depending on diseases. However, despite this systematic effort, around between 70 and 75% of patients remain without a genetic diagnosis. So therefore, we tried to tackle the challenge of solving these previously unsolved cases and to assess the use of exomiser for interpretation and its ability in incorporating the latest knowledge of diseases, genes, and phenotypes. A large-scale reanalysis of 24,000 unsolved cases from the 100,000 Genomes Project was performed using exomiser version 13. We therefore filtered for the rare protein coding segregating variants, and then we applied a conservative candidate selection procedure that ended up identifying 725 most likely diagnoses. This conservative candidate selection procedure includes many steps like the inclusion of de novo loss of function and pathogenic and likely pathogenic CLIN1 variant that were not reviewed by clinical scientists in the previous analysis, or as well as variant that have a human phenotype score more than 0.6 or were in green genes in the panels that was applied and it was following the correct mode of inheritance known for that gene. And then we were filtering for variants that were classified as pathogenic or likely pathogenic using the exomiser automated ACMG classifier. And latest, we were checking that the variant type, for example, missenses or loss of function, was consistent with the one previously reported in the literature for that gene, for that disease. And finally, we were checking, of course, that the phenotypic evidence for the patients were matching the one described for that disease in the literature. So finally, this set of 725 candidates were independently reviewed by the Genomics England Clinical Genetics team, which is composed both by clinical scientists and geneticists. And this led to the identification of new 463 novel diagnoses. So some of them, 153, remained as classified as variant of unknown significance, and some were rejected based on looking at IgV. So this is a little summary showing that the majority of these new diagnoses were based on ClinVar pathogenic, likely pathogenic variants. 144 of them were identified in the original 100,000 genome analysis. But they were overlooked as they affected genes that were outside the panel at the time of the analysis, while 107 were previously filtered out and retained here due to the Exomiser whitelisting feature, where such variants are always retained regardless of any filtering settings. Then we had the diagnosis, 98 of the diagnoses were made based on the de novo loss-of-function variant in genes that are present in the latest version of the panels. But actually 84 of them, for example, they were not in the panel when the primary analysis by Genomics England was run. Therefore suggesting that new evidence has since emerged for the disease gene association. Finally, Exomiser highlights candidates in genes that are still not present in the disease panel that was applied to the patient in 114 cases. So overall, this shows that 72% of the diagnosis involved the variant that were initially filtered in the primary or original 100,000 genomes analysis, but it was in the gene that was not in the relevant panel at the time of the analysis. But we also showed that for 205 of them, there were already evidence of this gene disease association in the literature. Therefore revealing the higher sensibility of a less targeted approach compared to the panel up strategy. And finally, the remaining were due to new discoveries that emerged after the original Genomics England analysis. So, however, this was very successful in terms of identifying novel diagnosis for 463 rare disease families. These analyses still required very extensive manual interpretation. And therefore, the next step for us was to try to develop analysis strategies to efficiently only highlight candidates due to new disease gene discoveries or newly designated pathogenic or likely pathogenic variants by ClinVar. So we tested and compared the ability of 7 releases of Exomiser databases from March 2019 until February 2022 to detect 37 real diagnoses from rare disease patients recruited to the 100,000 Genomes project, for which the diagnosis was in one of the 556 disease gene association that were firstly discovered in OMIM since February 2019. So from this data, we investigated in detail the combination of Exomiser scores that optimized the detection of these new diagnoses while still reducing the number of false positive candidates to investigate. So each variant called by Exomizer releases was classified as being a true positive, false negative, false positive, or true negative by comparing the results obtained using the combination of Exomizer score tested and the trusted external observation, which is the known genetic diagnosis. From these parameters about precision and recall, we extracted additional measurement like the F-score or the F2 scores, where in particular the F2 scores weights recall higher than precision. And the score combination with the highest F2 score was chosen, and we settled on a threshold of variance score of 0.8 and an increase in the human phenotype score more than 0.2 between Exomizer releases as an optimal way to detect candidates based on novel emerging evidence published in clinically relevant databases such as OMIM, Orphanet, or ClinVar. So over time, Exomizer also successfully reclassified this variant from a variant of unknown significant to pathogenic or likely pathogenic in 91.9 of the cases, following the implementation of the automated ACMG classification pipeline that was recently added to the latest Exomizer releases, and which I spoke about before. When combining the selected Exomizer score thresholds to the CMG classification, Exomizer actually shows very impressive results by drastically reducing the number of false positives, therefore increasing the precision to 87.5% with only a small drop in recall to 82.4%. So diesel analysis actually shows the ability of Exomizer to highlight and reclassify novel candidates for diagnosis over time in light of novel emerging evidence of gene disease association, and its ability to drastically reduce the number of candidates variants to review per case from a median of 30 variants to only 1 or 2 variants per case, therefore enabling efficient case reinterpretation in minutes rather than hours. So to wrap up today's seminar, we have explored the crucial and often complex process of variant prioritization. We explored the standard whole genome sequencing filtering pipeline for rare diseases and its associated challenges. We then saw a comparative overview of different phenotype-driven variant prioritization tools, highlighting their differences in capabilities and limitations. And finally, we introduced Exomizer, and we explored an example of real-world application by showing our recent interpretation project with the 100,000 Genomes Project data, where Exomizer plays a key role in solving previously undiagnosed rare disease cases. Thank you for joining me today, and I hope this seminar has provided a clear overview of phenotype-driven variant prioritization tools and the challenges associated with it. Thank you. Happy to take any questions.
Awesome. Thank you so much. This was a great presentation. Yeah, let's open it up if there's any questions, comments, thoughts.
I will start with a question. So this is a bit of a detail from your overview of all the other variant prioritization tools. You've put PPI and the model organism and I think pathway analysis in the same column. Is there any other tool other than Exomizer that uses cross-species data, or how many other tools are doing this? And if so, is there any other tool that is using cross-species data beyond mouse and fish?
Yeah, so if I go to that slide. One that I'm sure about is AI Marvel, which is surely integrating other model organism, but I'm not sure if this expands other than the mouse and zebrafish. Yeah.
AR Marvel's the one I remember.
Yeah.
Thank you.
Right.
Sorry, before we go on, I just wanted to acknowledge that Peter Robinson was a co-founder of the Exomizer software as well, like a key driver behind this whole project. So thanks to Peter. I don't think he's on the call today.
But can I just ask, just a curiosity, not super in line with your presentation, sorry, but like, so you had this clinician group go through 760 cases. How long did that take? Was it like they get back to you in 3 months or 3 years or?
Yeah, I think this was during COVID, kind
Oh.
of. So everyone was a little more free.
Yeah, there was one particularly keen person who helped a lot. And
Yeah.
But it's like a year-long process or?
it was like a few weeks. I seem to remember,
Okay.
trying to
Fair
remember.
enough. That's a lot less than I would have thought. Cool.
Yeah.
Very, very cool. Thanks.
Here, I'll speak for Daniel. He put in the chat, for the update that got about 90% F2 performance on the 100K undiagnosed cases, was that training on manually annotated cases?
Yeah, basically, sorry, the final exomizer update mentioned in the third. Yeah, so basically, what we use to measure F2 and the precision and recall is basically the definition of true positive, false negative. I come back to the slide. And this information about the actual trusted external observation that we use, it is kind of something that is annotated by genomic signals, or they are the officially diagnosed probants, and they give information about the variant that was diagnosed. So we use that as a trusted external observation. I hope I answered the question, but please feel free to ask me something further.
Daniel, do you mean the ACMG part from it? There's the graph with the So that was done as a the ACMG pipeline in Xmiser is basically just applying the rules that are in an algorithm manner that from the ACMG classification paper for sequence variants. So there's a whole bunch of them from the American College of Medical Geneticists. They came up to actually codify what people should use as a set of guidelines for classifying a variant as pathogenic or benign or not quite sure. So these are used in clinical practice. So this was added on as a sort of an extra layer on top of the Xmiser to actually label or tag the variants that come up with that.
So it wasn't an AI algorithm, essentially.
No,
Oh, okay. That's really impressive then to get that high level of performance on the manually annotated data. Well, I
Yeah, well,
guess,
it's kind of it's a bit of a circular thing because
yeah.
the people doing this should have also been using these same things. So these rules that are applied are applied in clinic or in the clinical practice to actually come up with the assessment of the variant that you have in there. I mean,
Now,
there are a set of guidelines, so they're not like totally fixed in stone as to what they are. But this is so that we maybe tweaked things here and there because Xamizer can't take into account all of the things that they can. But
yeah, that's my actual, if they're using the same rules, why is it not 100% then? What is
because
that
we don't
15%?
have available data that a manual curator can to do to maybe push something above or below a particular threshold. So that's because it's just an automated pipeline, so it can label things to an extent. So you can label categories as part of the ACMG classification. So you'll have a sort of say particular strength for a particular category, and these will get added up at the end of it. So there are some things that we can't add in there, which would be that they would be able to, like case counting, for example, if they've got X many cases that they have on this, we don't have that information available. So they could say, well, we've seen this in 25 other cases with exactly the same phenotype, exactly the same variant, and wherever else. So that gets added as another very strong evidence that this is the correct variant and it's a pathogenic variant. We don't have that information, so we can't use that one, for example. But yeah, this would be the kind of thing that we could train on a model with.
Thank you.
All right.
I will ask another question. So, are you expecting any major like paradigm shift in phenotype-driven variant prioritization in the next, let's say, 5 years or so in terms of technology now with the emerging AI approaches and so on? So, do you expect variant prioritization to look roughly the same as it does right now in 5 years, or do you expect it to look very different? What's your kind of like feeling for the development here?
Yeah, so I think Yasmin has already been testing out some the integration of some AI models into Exomizer, and thanks to Feeble, she's also able to test out the performances. As I would say that as usual, we are expecting AI to perform a lot better than previous methodologies, but so far we are experiencing similar results. Seeing how quickly AI is evolving, and hopefully my hope is that we will expand also with other new and better pathogenicity predictor to actually know more about the non-coding region and the pathogenicity or deleteriousness in those regions. I hope that we're gonna develop more in that area. Some of them are already, like CAD or RAM, are already spanning respectively non-coding and regulatory region. So hopefully we're just gonna expand on this.
Thanks.
Yeah, I think the main bit there is going to be the semantic similarity from embeddings and that kind of thing, which is the bit where we might get the most improvement from that kind of thing. I think what we've been asking with the LLMs directly to say, can you diagnose this particular set of phenotypes or link that to a particular gene or disease hasn't been particularly effective, as we've seen quite a few times. But I think we can actually use the embeddings for similarity matching within the Xamizer algorithm itself, which I think we can improve how things are working. Chris, you're talking. I don't know if you meant to be saying something. You've got your mic's. Your mic is off.
Yeah, and also with the LLM approaches so far, we've been basically using the LLM as an oracle. We've basically been saying, here's a list of phenotypes that give us your candidate for the disease. I think there's a lot more we could do with agent-based approaches, and where you essentially kind of cycle through each individual hypothesis, have it go off and do research, like read the latest papers on this disease, find if there's phenotypes that we haven't accounted for in our knowledge base, and maybe even run additional kind of like, you know, tools on the fly. So I think we're going to see a lot of activity in that area over the coming year.
Yeah, and actually maybe using that kind of agentic thing with linking to your own database of solved cases would allow you to access data that Exomizer currently doesn't have at all, because it's completely self-contained and has not much of anything else. But yeah, I mean, but then every time there's a new pathogenicity predictor, those things are very powerful in terms of what they can come up with. And so alpha missense, for example, has been one of the kind of the newer ones, which has been very successful on that front, which has been a completely new, it's all based off AlphaFold. So this is sort of de novo folding from first principles or whatever, which has never been sort of, this is a very novel way of predicting pathogenicity. But we can incorporate that into Exomizer as long as it sort of scales well with the other scores.
Awesome. Last call for any more comments, questions. This is great. All right. Well, thank you very much for being here, Letizia. You can find her on our Monarch Slack channel. And I'll put some information in the YouTube recording with links to Exomizer and things like that. So you should be able to find all you need to contact us if you're watching recording. So yeah, thanks so much, everyone. Thanks for being here.
Thank you.
Yeah, thanks, Leticia.